Hey, welcome to lesson three of Bio 205. So today we're going to actually be getting into some of the stuff that excites me the most about microbiology because we're going to be talking about a little bit of the history, which I love, and then also how microbes cause disease and what we can do about that. So I talked in lesson one a lot about spontaneous generation versus cell theory. So spontaneous generation for a long time was the understanding that living things arise from non-living matter. Essentially, maggots come from meat, mice come from grain, and disease comes from things that, smell bad, taste bad, look bad, things that we see and go, oh, I'm not going to eat that. I don't want to be around that. And that leads to this idea of miasma, and that flies and disease comes from that miasma around, uh, you know, dead things, decaying things, different matter that we find as human beings we don't want to be around just at a innate basic level. Now, this all changed when we first saw that cells were real and that experiments started to be done by different scientists to say, no, actually, cells are able to replicate and cause disease, and actually, specific microorganisms which of course are made up of cells, are able to replicate and cause specific diseases. So that gets into what we discussed with taxonomy and why it's important to say this specific bacteria or this specific archaea or this specific fungus or parasite is that thing because we can treat that exact thing for the most part. Now, there's something called Reddy's test of the visible, which was used in around 1668. And the question was, do maggots spontaneously appear or do they come from flies? So the design was to have an open jar with meat that was exposed to flies, a cork sealed jar, so no flies could get into it, and then a gauze covered jar. So just a thin layer of fabric over the jar, which allowed air but not flies. So we have an open jar, two flies and air, a cork sealed jar, so no flies, no air, and then a gauze covered jar, which allowed air in and flies in. The results were that the maggots only came from the meat in the open jar, not on the gauze jar and not on the corked jar. So this was kind of the first indication in the mid 1660s to late 1660s that actually spontaneous generation isn't what happens. There's something that happens where flies lead to maggots and then maggots lead to flies, right? So there's a parent-child relationship happening here. It doesn't spontaneously come out of the meat. Then there's a battle between Needham and Spallanzani in the mid to late 1740s to 1760s. So John Needham boiled beef broth, then sealed the broth, and the broth turned cloudy. The interpretation here is that there is this spontaneous generation, um, but there's a lot of flaws with this experiment. It turned cloudy not because he killed everything in it and then sealed it perfectly under aseptic conditions. It turned cloudy because he boiled it and then poured it into a jar, which wasn't aseptic and was exposed to air, and then capped it with a cap that was not aseptic. So this is one of those experiments that back then kind of made sense. You don't know where microbes are and how they're everywhere. But the way you did the experiment, of course, we now thinking about it, know that you're going to have some sort of growth in that. It's like if you boil soup and then pour it into a bowl and then cover the bowl with aluminum foil. Yeah, it's going to go bad. We just know that because microbes are everywhere and they're going to turn it into horrible fetid soup. Now, Spallanzani boiled the broth longer, sealed it during the heating, and then also paired that with an open flask. So the difference here is that he made sure that everything involved was extremely well boiled and therefore would have killed all the microbes. So the jar that was used, the broth that was used, the cap that was used. Was his aseptic technique perfect? Probably not, but it stayed sealed and was not cloudy for a longer period of time. So it stayed clear, whereas the open flask grew microbes. So there's this counter thought going on where someone says, well, I did that same experiment and I got these results. And the other person says, no, your experiment was poorly thought out. I did a better version of it and I got these results. And then we have Pasteur come along with a swan neck flask. So a normal flask is it goes up and is just open here to the air. A swan neck flask, it goes and then it curves around. And the reason that curvature is important is most microbes are not motile. They cannot move around easily. So they might drop into part of the flask, but they can't go up and around all the glass. And that is the kind of key to this experiment that Pasteur performed. So that swan neck shape, it lets in less air and it traps all the dust and microbes in its curves. The result is the broth is sterile and remains sterile if you boil both the broth and the flask when the neck is intact. Tipping the broth into the neck, so actually moving the broth into the neck and then back down, leads to growth and leads to cloudiness. And that's how Pasteur concluded that air doesn't generate life. It's the microbes. It's the microbes in the air and in the dust that lead to this contamination, sort of sealing the final nail in the coffin on spontaneous generation, and instead leading to this microbe and life begets life thought and theory, and of course, at the end of the day, truth of when life is generated, it always comes from previous life. Now, there's the initial generation of life, which there's a lot of debate on how that happened and could happen and might happen again, and how we could do it in a lab. But we're not going to worry about that. For the course and for this, we know that all life now comes from life before. And that's what Pasteur definitively proved and why that experiment was so important. Now, uh, there's this thought of sterilization and sterilization being permanent. And that's the other thing that Pasteur shows as a side effect of what he's doing, that you can sterilize something, but as soon as you contaminate it, so pouring the sterile liquid into the neck of the flask and then back down, that contaminates it. So you can have something that's sterile get easily contaminated, and that's why these other previous experiments did not work. Um, there is a lot of this in modern day. So sterilizing things and then preventing contamination. If you think about a surgeon who's getting scrubbed up, they wash their arms for a long time. They're scrubbing under their nails as much as they can, and then they put all of the sterile items on. They actually have other people come in and help them to get dressed who have already gone through that same thing, put on sterile items, and then they are putting on the other sterile items onto the surgeon who's performing the surgery to mitigate the risk of contamination. And we do this because we know that nothing can really ever be perfectly sterilized. If you think even about you have canned food that is boiled, it's put in a can, the process is very, very, very good at sterilizing that food and keeping it sterile for a long, long, long period of time. And a lot of the times that works for years and years and years. There's people that, will crack open old cans that they find from the 30s and 40s and eat what's inside, which is a little wild to me, but they'll eat what's inside and it's perfectly fine. And that's because that works. But if there is any contamination that gets in there or enough time passes where the slightest little amount of microbial contamination gets in there, you will have a failure of that system. And that's something we need to be aware of, especially in the medical community, of people can say, oh, well, I sterilized everything, but that 99.999% is not 100% sterilization. So there's always going to be some bacteria, some viruses that exist. Luckily, we have immune systems to deal with that. But when you think about something being sterilized, you just need to know that there is likely some small amount of contamination that goes along with that. It's very important in the medical community to be aware of and then take precautions against. So when we came to this theory that spontaneous generation is out the window and we have what's called biogenesis, so life becoming or growing out of previous life that existed. So one cell begets another cell. You know, you have two parents that beget you, life comes from life. Then we get cell theory and the germ theory of disease. So cell theory is the basis that life comes from these individual units called cells. We see it in every single domain of life, so that's bacteria, archaea, and eukaryotes. Cells are the basis of what we call life. Viruses we'll get into later, but think about cells equal life for now. And then we get germ theory of disease. So germ theory is that, well, we also get disease from these cells. And that, in turn, is very important for how we tackle disease and treat disease. Now, the development of cell theory happened over a long course of time. Again, in the late 1660s, Hooke names the cell based on, looking at a cork under a microscope. And, that starts this whole conversation around cells. That's where the name comes from. Then von Leeuwenhoek sees living cells in the 1670s and 80s with a simple compound microscope. So that's two lenses for the simple compound microscope. And he sees what he calls animalcules, and that is cells that are living. So this is your bacteria, your protozoa, your archaea. He's seeing those things, and it opens this entire world of life at a microscopic cell scale. Then it goes from this observation into this theory. Remember, theories, as we talk about them in our day-to-day life, are different than scientific theories. Scientific theories are something that is, with all the data we have, a proven reality. So the theory of relativity in physics is something that is a basis of a lot of different physics that we do every single day. It's accepted. The same thing with this theory of biogenesis and the theory of the cell. It develops over time, though, because there's new information that you add into that. So theories are very close to a truth, but they can be informed by additional information. And that's in science versus in day to day, you talk about theory like, well, I have a theory that, this person likes this person based on this evidence, and that is just kind of your own thoughts, your own inferences, whereas science is based on cold, hard observed, categorized facts. So that differentiation is very important. So scientific theory is very different from your day-to-day usage of the word theory. And the theory of biogenesis is supported first by Hooke finding cells, then von Leeuwenhoek seeing these animalcules, and then a bunch of different observations. So there's a person called Matthias Sheldon. Sheldon. It's a German name. Sorry, I'm not great with that pronunciation. And he found that plants are made up of cells. And then also in this 1800s, start of the 1800s period, there's Theodor Schwann, and he found that animals are made out of cells. Together, all these organisms are seen to be cellular. So this cellular basis, this cellular generation of life as a whole from this basic one unit is this theory that is established. And then there becomes the question, well, where do cells come from? And that was unknown for a while until Rudolf Virchow discovered that cells can arise from pre-existing cells only. And this is when you have a cell that then splits into two. And this happens in bacteria. Bacteria will split into two cells. And then, when you are a original entity, I guess is the way I'll phrase it. You have a sperm enters an egg, and then the egg starts to divide, and those cells divide and divide and divide and divide, and that ultimately becomes a person. So it's the same overall happening, the same occurrence of a cell divides into another cell. There are more details there, there are weird differences, but again, that's the overall observation and information that's put into this theory of biogenesis. Now, the modern cell theory, of course, theories can change over time as new evidence comes up, is a little bit more detailed with a couple asterisks put in places. So you have an egg and you have a sperm, and they come together and they make one cell. So that's something that, it doesn't quite fit that earliest theory of, well, a cell divides into two. There's other inputs happening here. And that's why you have half of your mother and father's genetic code, because those two cells are only one half. And when they come together, they make a whole, and then that starts to divide. So that's something that was added later to this theory. But what hasn't changed is all things are composed of one or more cells. The cell is the fundamental base structure of a functional organism. Now, functional organism is an interesting term here. The reason it's used is because a virus could technically be an organism depending on how you talk about it and classify it. They're in this weird point where they have to parasitically infect a cell to replicate and live. Well, they can't function on their own with their own structure, and that's why this terminology is used. So, is a the cell is a fundamental unit of a structured and function of an organism. And that's why it's used, because you have viruses over here doing weird things. All cells arise from pre-existing cells, not from one pre-existing cell. So that's another modern differentiation from cell division. But remember, it's not just one cell, it could be multiple cells coming together, the egg, the sperm, and that is what results in additional cells. Um, Cells generally carry genetic information. This is passed on during cell division, and this is another, modern take because they didn't know about DNA back in the 1800s when this theory was first proposed. So then we get to another theory, the germ theory of disease. So early outbreaks, again, they had that whole miasma thought. It's not microbes. They haven't been linked to disease. It's this miasma that's around dead, decaying things, sick people, etc. Um, there is one particular person that was kind of key to this, and that's Samuel Weiss. He worked in a maternity ward. So back in the day, and this is kind of iffy when you know anything about how disease works, doctors would do autopsies, and then they would go treat sick patients, and then they would go deliver babies, all in this same ward. There's not a lot of doctors, so they're doing a lot of things, and they're not washing their hands. There's actually a interesting thing with surgeons. When surgery was first developed as a discipline, it was a badge of honor to wear your surgeon's smock and never clean it because it being dirty and gross with all of the viscera of all your surgeries was a badge of honor that you had done that. There's actually uh, in a speech to an early surgical class, there's a line that's been noted down that I think is incredibly interesting about how a lot of surgeons, during the course of learning to be surgeons, would die. It would be a huge percentage of the class because you're dealing with people who are infected, people who are having all these issues. You're, getting your blood on their blood on you, your blood on them. So you're very likely to get infections and die from those infections. That's something that happened in these surgical schools in the 1800s because we didn't understand disease. It was coming out at the same time that surgeries were being popularized, right? So that's an absolutely crazy history lesson right there. But Samuel Wise develops this thought of, well, I'm going to wash my hands. I'm going to get all this blood and other stuff off and then deliver babies. And what was seen is that this simple hand washing meant that more babies lived in his ward than all the other wards in the other hospitals during the time period. We were talking about surgeries just now. There's Lister. He develops aseptic techniques in surgery to reduce infections, not just for the surgeon infecting the patient, but the patient infecting the surgeon, and reducing those deaths of surgeons due to complications from their patients. There's a famous uh, event, I'll say, that a surgery had a 300 time or a 300% mortality rate. So the surgeon was doing a surgery, ended up killing the patient. Someone in the observation died of a heart attack, and then he infected a nurse by accidentally cutting her, I believe, is the way it worked out, and she got an infection and died. So stuff like that was commonplace. It was accepted until Lister said, "Why are we doing it like this? Let's do it a different, better way." Um, Koch has Koch's postulates, which connected, again, specific microbes to specific diseases like anthrax and tuberculosis. So he was also instrumental during this time of connecting, hey, it's not just all of these nasty things creating all of these other nasty things. It's one disease creates, or one microbe creates a specific disease. So there's another man in London, Snow, who traced cholera outbreaks to contaminated water. So they're having these cholera outbreaks. He was the first progenitor of public health. So he looked at the water, looked at it under a microscope, and said, "Oh my gosh, we have cholera in all of these water sources. No wonder people are getting sick. Let's do something about this." And that is the generation of public health from this one person going out and finding the source of the disease. So all of these people together were instrumental in establishing the current understanding and the current disciplines that we use every single day around microbes. We're gonna quickly talk about prokaryotes and eukaryotes and how they're different. So prokaryotes, they're often smaller, no nucleus, often single cellular, have specific ribosomes, and all of these other structural elements that aren't really found on eukaryotes. Eukaryotes are often larger. They have a nucleus with DNA. That's the defining factor of a eukaryote is it has this nucleus, this structure. They have a lot of different organelles that bacteria typically don't have, like the ER, the endoplasmic reticulum, the Golgi. They have mitochondria or they have chloroplasts, and they also have oftentimes a different kind of ribosome. So there's a lot of differences between the two, but again, like I talked about in lesson one, there's a lot overlap. We put them into these buckets, but really there's a lot of overlap. It's a Venn diagram. Now, we're going to talk about these different organelles and a little bit about the importance of all of them. It's very important to understand what the different organelles are, what their function is. So we're going to start with the nucleus. So or that is the region in which, for eukaryotes, all of the chromosome is packaged. And in prokaryotes, we have something called the nucleoid. So, nucleus is in eukaryotes, where all the chromosomes are packaged. We have the nucleoid in prokaryotes, and in that they have this concentrated region of DNA, but it's not bound inside of its own membrane, inside its own organelle. And that's the difference there. Ribosomes are in both. They're responsible for protein synthesis. So taking that genetic code and translating it into a actual protein that is a functional unit inside the cell. There are different inclusions in prokaryotes. These stored nutrients are responsible for stuff like buoyancy if they're in water or in your blood even. So they can do weird things to move around in that way. Again, they are going to have, and this is true for both prokaryotes and eukaryotes, some sort of membrane. This is semi-permeable. Think about it as a sea with a bunch of islands in it. The islands are these concentrated regions of proteins that are talking to the outside and then translating that to the inside. And then you have this undulating membrane that is going to be able to be passed through in certain areas, whether it's transported through those islands and all that communication machinery, or it's able to just penetrate through the membrane as is. Now, We have a membrane, but we also have something called a cell wall. And this is going to be in plants and in certain bacteria. You'll have a cell wall. It's a thicker, less permeable layer, is what I'll say. So this is very important because a lot of antibiotics will target the cell wall and lock that up, prevent bacteria from using that machinery on the cell wall from communicating and dividing and things like that. We already talked about gram-positive versus gram-negative bacteria, so this kind of goes along with those stains, so having a cell wall or not having a cell wall. A lot of bacteria will fall into one of two morphologies. Now we're getting outside of the basic organelles and we're going into the overall structure of the bacteria. And this is typically in two different shapes. You have your rods and your spheres. So rods are known as bacillus, so they're rod-shaped bacteria, they're long and thin, or you have coccuses, which are spherical. So if you're looking at these on a slide, you don't need a stain to tell you it's a rod. You can just see that. And then you need to stain to see if that rod is gram positive or gram negative, etc. So the two main differentiators that we look at first are, is it a bacillus, a rod, or is it a coccus, a sphere? And these, of course, have all sorts of arrangements. It could be a sphere by itself. It could be a rod by itself. It could be a rod in a chain or a sphere in a chain. It could be in clusters and all these other things. So those also have distinct names. And this distinct morphology, again, can inform us what kind of antibiotics to use, what kind of organism this is. Is it, a bacillus? So the rods, in a chain of rods, which is a streptobacillus. So strep throat, you know, might be related, but also there's spheres, so coccuses that can be in chains, and those are called streptococcus. So you have streptobacillus, streptococcus. I'll be honest, I forget off the top of my head which strep throat is, but it's one of those two. It's a chain of bacteria, and you can look at that and just see it under a microscope. So when you're thinking about, okay, how do I categorize these? You're going to look at the morphology. Is it a rod or a sphere? And then you're going to look at the arrangement. Is it, in a chain? Is it in a clump? Is it simply two right next to each other? And that's your first indication of what kind of microbe you're dealing with, at least when it is a prokaryote. Now, you can get into a bunch of different structures that are associated with these bacteria and that are associated with the pathogenicity of the bacteria. So, when we're talking about that, I'll give some examples, but just know there are always more examples. That's one of the great things and one of the kind of pains about life and science is that you have some great examples, but there's overlap and then there's outliers that you're dealing with at all times. So, one example is you have flagella. This makes bacteria motile, so it can move around more easily, and that is on a lot of different bacteria that are pathogenic. So it can move around, which means it can get places more easily, find the cells that it wants to infect or eat, the different, metabolites that it wants to eat, it can get there. So these can be very dangerous because they're not relying on your body's own systems to move them around. There's something called mycolic acid, which is this waxy coat that goes over things that are mycobacterium. So Mycobacterium tuberculosis, TB, has this and it can be seen with an acid fast stain. We talked about that in the last lesson. But it has this waxy coat which protects it from certain things getting in there and degrading it, right? So there's many examples of these. Now we're going to talk a little bit more about eukaryotic cell structures, and this is actually a lot easier to discuss because bacteria are so diverse in which cell structures they have. It's really comes down to, is it, a cell wall or not? So is it going to have a cell wall be gram positive, gram negative, or is it going to have these higher level structures? Is it a coccus or not? Right? And all the internal organelles can change depending on what bacteria you're looking at. So we use those broader terms when we're looking at bacteria to define them, and then we use certain stains to see further differentiators for clinical application. But eukaryotic cells, they have a lot more machinery, and that machinery is more standard. So we have a nucleus. That nucleus is going to be this membrane that surrounds all your DNA. It's going to store your DNA and then sort of meter out your access to that. Why is this important? Well, you have viruses that come in and they say, we would like your DNA, please, because we need that to replicate. And it is another barrier to essentially protect you from these outside infections getting into your cell and using your own machinery against you. There is then the endoplasmic reticulum. So this is separated into the rough and smooth. So the rough endoplasmic reticulum and your Golgi are going to be in charge of protein synthesis and modification. So there's a lot of detail there. There's a ton of detail there. Just remember, rough ER and Golgi are responsible for protein synthesis and modification. In addition to this, they're also responsible for the secretion of vesicles. So vesicles are membranes that bud off. So these are going to come out of your rough ER and Golgi. They're going to be these membranes that bud off of those organelles and are transported around. Why is this important? Well, some proteins you only want specific places. You don't want proteins that are responsible for marking and degrading viral, you know, capsids that are inside of your cell to accidentally attach to your nucleus, for example. That's an extreme example, but I think it's a good one. So you're really making proteins, modifying them, and then shipping them other places with the rough ER and the Golgi. Now, of course, because scientists love naming things terribly, there's also the smooth ER, the smooth endoplasmic reticulum, and that's responsible for lipid synthesis. So lipids um, are fats that are going to go to your membranes, to all the different membranes. And that's all you need to remember right now for the smooth ER. Of course, you have the mitochondria, the powerhouse of the cell. This is responsible for what we call cellular respiration, for energy conversion within the cell. If we didn't have mitochondria, we would not be able to have cell activity and we would, as an organism, die. So it's very important for that. In plants, though, so this is a differentiator. In plants and algae, they have chloroplasts, which are responsible for photosynthesis, which turns light energy. So from the sun, you get the photons, they excite part of the chloroplast machinery, and that ultimately produces energy. So they don't have mitochondria, they have chloroplasts instead. Chloroplasts are green, and that's why plants are green. They're actually green because that is the highest energy peak in the visual spectrum. So they get more energy when they're green, and that's why they turn red and yellow and everything in the fall because they're losing leaves are losing those chloroplasts and then turning red and then ultimately losing their leaves because they are going dormant for winter. So just two major differences there. We and a lot of other eukaryotes, animals, just think of all animals, have mitochondria for energy, and then plants and algae have chloroplasts for energy. Now, there's something else called a cytoskeleton. This is filaments that run along your intercellular space. And these filaments are responsible for the shuttling of different things. If you think about how does something get somewhere inside of a cell? As this gooey mess of water and proteins and lipids and vesicles all floating around, you would think, oh, well, it has to have a flagellum to like wiggle through and swim through that. But we don't have those. So what do we do on a smaller scale, on that protein scale, on that lipid scale, where you can't have a flagellum swim around for you? Well, we have all these highways that are these filaments that certain proteins will grab another protein and then attach to the filament and then literally walk from one side to the other. So they're these shuttle lanes, similar to like a bullet train or a tunnel or an airplane. It's specific lanes that they're traveling along to get whatever unit, whether it's a protein or a lipid or an entire vesicle, where it needs to go. And that's ultimately called the cytoskeleton. This is one of those things that's a lot easier with pictures. So I'm saying a lot of this. It would probably be best to look at pictures, listen to this, and then look at the pictures again to kind of get the full understanding of how these things work, where they are. And that will sum up the main cell structures for eukaryotes. Again, prokaryotes, they have a lot of different cell structures that are very specific to the type of bacteria that they are. And that's good for us because it allows us to classify them easily. It's bad for understanding when you're first jumping into this of, okay, but like what is in a prokaryote? It's going to be different in a lot of cases. So just know that and review those main differences, understand what's in typically in eukaryotic cells and the main differences between the eukaryotes, what's there, and the prokaryotes and what's there. Archaea, again, there's a lot of overlap with these things, especially into prokaryotes. And they're not studied as much. They're not taught as much. So we're kind of just closing the book on them for now and setting them aside because prokaryotes are already diverse in the different organelles that they have. Archaea are even more diverse. So when you get down the line and you're studying them, all of these different things come up where you're like, wait, how do we categorize this? How do we tell the difference here and there? And that's a bit beyond the scope of this lesson. So just focus on eukaryotes, prokaryotes, the history and why it's so important for these differentiations to have been made. And then you'll be in a pretty good place for understanding everything else that's going to come in the course. So thank you so much for sitting through what was half history lecture, half a lot of detail about cells and their differences and why it's important. And please, as always, let me know if you have any questions. Thanks.